Dear students, welcome to the lab session gram-negative bacilli. This includes Pseudomonas, Brucella, Haemophilus, Vibrios, and Aeromonas, Campylobacters, and Helicobacter. In this session, we are going to discuss the main species and their diagnostic laboratory tests, highlighting the oxidase test, and also identification of species under each of these genera. As you may still remember, in a gram stain, we have the gram-negative bacilli, and these, based on the growth of MacConkey, could be positive or negative. And in this lab session, we're going to discuss these MacConkey negative, Haemophilus, Brucella, Vibrio, Campylobacter, Helicobacter. And also in the MacConkey positive, we have the Pseudomonas and Enterobacteriaceae. And oxidase test is very important to tell them apart, keeping in mind that one of the members of the Enterobacteriaceae, which is called Plesiomonas, is also oxidase positive. So for Pseudomonas, we have a single species called Pseudomonas aeruginosa. The habitat in moist areas in hospitals, which is also a major source of nosocomial infection. It colonizes human and saprophytic organisms. And beside being nosocomial, they are associated with burns and wounds. It's also a key pathogen in cystic fibrosis and infections in immunocompromised subjects, as you can see in these images here. What is characteristic about Pseudomonas? It's a strict aerobe and can be seen in infections in cystic fibrosis in the lung or in the skin, direct contact with the oxygen. There is what is here important to see this bluish or greenish discoloration, which is related to the big pigments released by Pseudomonas. And this discoloration or these pigments are highly suggestive of Pseudomonas infection. Direct method is by the gram stain, and they are gram-negative bacilli. In the cultivation, they are MacConkey positive. They can grow on blood agar, as you can see here. They are also beta hemolysis. And also they can grow on Müller-Hinton, to see the different pigments there, and also on triptychoid soy agar, TSA. And they are, as we said, oxidase positive. And in TSI, triple sugar iron test, they show alkaline and no change reaction in the butt. They are also characterized by the production of these bright bluish biocyanin or green pyoverdin or red pyorubin or brown pyomelanin diffusible pigments. And they can best be seen on Müller-Hinton agar, MH, or the TSA. Here, this is the, for example, green discolory or pigmentation seen on the TSA slats. Now, oxidase test is the test that detects the cytochrome activity in strict aerobes. And usually this is done by applying the oxidase reagent, which is 1% of the Covax substrate, and using a platinum wire loop or a wooden stick, we can take a few colonies from the culture, preferably not older than 24 hours, and then rub it on the moist filter paper where the reagent was applied, and we wait for 10 seconds. Within 10 seconds, if there will be a change, a color change to deep blue or purple, then it's positive. If there is no change, then it is negative. So as you can see here, this is the positive one, and this is the negative. Now we go to the other genus, the Brucella, and we have 4 medically important species. And in this table, we see the preferred animal host and the number of biovars. What is special about Brucella, they are intracellular and is responsible for the also what is called the Malta fever, and they are non-motile, fastidious, and gram-negative Bacilli. All these species except the Brucella suis they are considered as potential agents of bioterrorism. And brucellosis is the most commonly reported laboratory-acquired bacterial disease. It is usually, the disease is caused by unpasteurized milk and dairy products, in particular cheese, are common vehicle to transmit the disease of Malta fever, and the infective dose as low as 100 microorganisms can cause brucellosis. For diagnostic laboratory tests of the brucellosis, there is no widely acceptable direct detection method. Cultivation is difficult, and since this is difficult, then we have no ability to do antimicrobial susceptibility testing. And so what remains is the serodiagnosis by taking serum, and it is the widely used method for diagnosis. And we do serum agglutination test, and the titer of 1 to 160 is considered diagnostic if it agrees and consistent with the clinical picture and epidemiological findings. Of course, Iraq is endemic for brucellosis and Malta fever. What is seen as limitation in the SAT test that does not detect the Brucella suis antibodies. Another test is the indirect Coombs test, can be performed to detect the non-agglutinating or incomplete antibodies in the complicated or chronic cases of brucellosis. Additionally, we can have the ELISA test, which can be particularly useful in case of neurobrucellosis. The other genus in this group is the Haemophilus, and we have the H. influenzae, Haemophilus influenzae, as a normal flora in the upper respiratory tract. It can be responsible for meningitis or pneumonia. On the other hand, we have Haemophilus ducreyi, and this species is not part of a normal flora, but a pathogen, and is responsible to what is called the chancroid, which is a sexually transmitted disease, and is different from the chancre that is remarkable of the syphilis disease. And we have other Haemophilus species as part of a normal flora in the upper respiratory tract, and they are considered as opportunists. It means they cause infection only when there is a suppression of the immune system in that individual. In general, Haemophilus species are fastidious microbes, and they require either the X hemin or V nicotine adenine dinucleotide, the NAD factors, or sometimes both. And they are required, both of them are required, for example, as in the case with H. influenzae to grow. H. influenzae can be subdivided into typable or untypable. The typable depends on the presence of the capsule, and they can be subtyped into A to F types, or non-typable strains. The one responsible or the most common pathogen in human is the H. influenzae type B. Now, for the diagnosis, we have special notes about specimen collection. It should be transported to the appropriate media as soon as possible, because Haemophilus are very sensitive and susceptible to drying and temperature extremes. Also, what is important about collecting specimen, they should be done through bronchoscopy, through what is called a bronchoalveolar lavage, which is used in this case. In the bronchoscopy is inserted, and to take a specimen from lower respiratory tract, and not to mix the specimen with the normal flora that is present in the upper respiratory tract along the way where the bronchoscope is inserted, higher up, okay, in the respiratory tract. So this way we can only take specimens and reflect the disease rather than being contaminated with the whole microbes in the upper respiratory tract. Now, in case of Haemophilus ducreyi, which is responsible for the chancroid, this is in this image. You can see the chancroid. It's very important to take this specimen from the base of the ulcer, not from the margin. And the swab should be immediately, within 10 minutes, plated to the selective media to maximize the chance of recovering Haemophilus ducreyi, the causative agent. Now, direct detection method, we can see the gram-negative bacilli here, as you can see. And also, it depends on the site of infection. If meningitis, we take CSF. And in order to increase the possibility of recovering the microorganism, we do centrifugation, which is called concentration technique. In this technique, we can do centrifugation of the CSF at 10,000 G for 10 minutes. This way, we're going to take the organism from the sediment and do gram stain. In the gram stain, if it is not possible to see the organism, we can do acridine orange stain, which is a fluorescent dye. And by fluorescent microscope, we can have more sensitivity. The human eye can better detect the microbe using the fluorescent dye because of its higher sensitivity. Okay. There is also the latex agglutination test to detect the capsular antigen, and we said that H. influenzae type B is the most common pathogen. It is sensitive and specific to detect the H. influenzae B, even if the patient was on antibiotics, which is very helpful. And false positive results can arise if there is a recent immunization against H. influenzae type B. Now, there is also the molecular techniques using PCR. In this case, there is the advantage of absence of cross-reaction or autoagglutination, which are non-issues to occur in serotyping. And pulse field gel electrophoresis is the gold standard for typing of Haemophilus different species. Now, H. influenzae, as we said before, they are fastidious, and they require the growth of both, or the presence of both X and V factors, the Hemin and NAD factors. And both of these factors can be found in the chocolate agar, not on the blood agar. And for the because in the blood agar we only have the X but not the V factor. In chocolate, on the other hand, we have both of them. So chocolate is used to grow H. influenzae. And the growth is enhanced under 5 to 10% CO2. And you may still remember we use the CO2, the candle jar for that at 35 to 37 degrees centigrade for a maximum of 72 hours for 3 days. Okay. Now there is what is called the satellite phenomena. In this phenomena, there will be growth of H. influenzae around beta hemolytic staphylococci species on blood agar. As you can see here in this image, this is a blood agar, and the white colonies are the white large colonies are actually staphylococci. They are beta hemolytic staphylococci. And here we can see at the arrowhead, we can see the small colonies are only seen around the beta hemolytic staphylococci, not all other staphylococci which are not beta hemolytic. Why is that? Because by hemolysis we can now have the V factor the V factor, because the X factor is already available in the blood. But because of hemolysis, now we have the V factor, and together with the X factor, they are both required by H. influenzae to grow. And this is the reason why we see the H. influenzae colonies, small colonies, only there where the hemolysis occurs. Okay. Now we can do serotyping for H. influenzae. This is to identify the B strains. And also the H. influenzae shows negative porphyrin test. For H. ducreyi, they require special media, and they require only the presence of the X factor. So it can be grown on Müller-Hinton based chocolate agar supplemented with 1%. and this is the X factor, plus the vancomycin and brain heart infusion based agar supplemented with 10% fetal bovine serum and vancomycin. So this is the special media, and growth additionally requires the high humidity under CO2 condition for as long as 7 days. And Haemophilus species, being fastidious, they don't grow on MacConkey agar. Now we go to Vibrios and aeromonas. The one species that is responsible for the cholera epidemic and pandemic is the Vibrio cholerae, but not all. Only the serogroup 01 and 0139, because we have about 206 O antigens, somatic antigens, but only two, the 01 and 0139, are responsible for the epidemic and pandemic, for the cholera. And all of the O antigens, they cause cholera-like diarrhea or extraintestinal infection. For parahemolyticals, they are responsible for gastroenteritis or extraintestinal infection again. Aeromonas species, they are mentioned here because they can be easily mistaken for a cholera, and due to the misleading results they present in the lab. They are responsible for gastroenteritis, wound infections, and endocarditis. And the usual habitat for Vibrios is the aquatic environment. They are grow at best in alkaline pH, and they are going to be killed by acidic end products or acidity. Usually, when we try to take the specimen from the stool, usually the clinical manifestation of the cholera, the patient is presented with dehydration and also with painless, rice, watery, afebrile diarrhea. And there should be a good transport medium for the stool samples in cases suspected of cholera. And the name of the transport medium is called the Carey-Blair medium. Why is that? Because to protect the microorganism, because it's sensitive. And also we do ELISA test, latex agglutination test for Vibrio cholera toxin, which is the direct cause for cholera infection. And the gram stain, we can show here the gram negative, straight or slightly curved comma-shaped bacilli. The aeromonas can also show the same appearance and shape, and they can be also gram negative, straight rods with rounded ends or cocoa bacilli. So here, this is the comma shape of Vibrios. You can see here very clear. There is no direct detection method for aeromonas species now available for laboratory testing. Now, a special medium or selective medium for Vibrio is called the TCBS, and these letters stand for thiosulfate and citrate bile salt sucrose. From the name, you can guess that it is a selective for Vibrios and contains 1% NaCl. Bile salts, from the name, they are important to inhibit the growth of gram-positive organisms. Okay. If you may still remember, this is the same thing in MacConkey agar. We have also bile salts to prevent the growth of gram-positive. And there is high pH, about 8.6, which inhibits, again, the growth of other intestinal flora. And it is differential. This media is also not only selective, but differential because of presence of sucrose. And typically, colonies of Vibrio cholerae will produce yellow color because of the sucrose fermentation, and which does not take place in case of Vibrio parahemolytica. So if you see this pigmentation or yellow color, it means this is Vibrio cholerae, not parahemolyticus. And the pH indicator is the bromothiamyl blue and thiamyl blue are used for these type of media. The cultivation is done by incubation at 35 in ambient air. and they can grow on MacConkey agar as lactose or non-lactose fermenters. There will be variable beta hemolysis on the blade agar. The alkaline peptone water is an enrichment broth, and the alkaline is 8.4 to preserve the Vibrios. And this should be done incubated for 5 to 8 hours from the stool specimen in this enrichment broth. And then after this enrichment, there will be selection for the group of favored microorganisms or desired pathogens while getting rid of all other normal flora. And then after that, we can do subculture on a selective medium like TCBS. The Aeromonas agar is selective for Aeromonas and contains the D-xylose as differential characteristic. And Aeromonas and Vibrios, both of them are oxidase test positive. And it's very important to notice that we shouldn't take the culture from media that contains fermentative sugar, like sucrose or any other sugar. Why? Because after fermentation, the end products of fermentation will be acidic products, and this will lead to low pH. And we said before that Vibrios are very sensitive to acidity, and actually they are going to be killed by this acidity, making oxidase test not that useful. For the biochemical test, we can use the EPI system, EPI20E for enterobacteriaceae, from Biomirio for Vibrios. Okay, and serogrouping, serotyping of Vibrios can be based on the presence of the different somatic O antigens, and we said there are about 206, but only O1 and O139 responsible for the color. Now, in this table, you see the different tests that can be performed additionally in order to differentiate between Vibrios and Aeromonas species. Because why? Because in outbreak, for example, the outbreak that happened here in Iraq, some cases were labeled cholera, where actually they were Aeromonas infection. So in order to tell them apart, we use the string test here in this image, using this wooden stick, and the using 0.5 sodium deoxycholate, and we put the microorganism there. If you apply the stick, if there is like this string happened because of the mucus nature, then it is positive, and this indicates the Vibrio infection, not Aeromonas. Again, the growth on TCBS is only positive for Vibrio, but not Aeromonas. And the tolerance of 6% salt is only seen in Vibrios, but not in Aeromonas, which are intolerant to that percentage of salt. So in this image, we see this positive string test, okay, and in order to identify color. Now we go to another species, or two general Helicobacters and Helicobacter. The Campylobacter, we have Campylobacter jejuni, responsible for gastroenteritis, as well as Campylobacter coli. But in jejuni, the gastroenteritis can progress to a certain neurological disorder or a syndrome called Guillain-Barré syndrome, which is considered as a neurological emergency case. It can be very deteriorating within a short period of time, and it is defined as an ascending muscle weakness or paralysis. And the patient can end up with having difficulty in swallowing or breathing, and respiratory arrest and death. So it is considered as an emergency and should be dealt with immediately. And what is important to notice about Campylobacter, they are invasive infections. Therefore, we can see the RBCs and pus in the stool, which is not the case, for example, in the Vibrios and cholera, because it is considered non-invasive. Now, another microorganism which is present in the stomach as a natural habitat for it is the stomach, particularly in the area of the antrum, It is the Helicobacter pylori. It is associated with gastritis, peptic ulcer, and gastric cancer. However, association does not mean causation. It is non-invasive and it's mucosal, and it can lead to can be associated with different pathologies depending on the clinical manifestation. So for the direct methods of detection in the gram stain stool sample, we see small seagull winged gram-negative bacilli for Campylobacter. And also we can have PCR detection or enzyme immunoassay in the stool. And this is the seagull winged here, as you can see, this microscope slide. And also the cultivation method for Campylobacter, we have the Campy-BAP and Campy-medium CFA are selective for Campylobacter. And in order to maximize recovery, we use two selective media at two incubation temperatures, once at 32 and once at 37. Why is that? Because the Campylobacter fetus, which is another micro-opportunist, can be overlooked if the temperature was not set to 37. So we set two different temperatures for the selective media in order to recover Campylobacter fetus. And the Campylobacter are both oxidase and catalase positive. And for the lab report, it is usually enough to mention it is the Campylobacter species and no need for speciation, means to mention the species and identify the species, because the treatment is the same, and most of the cases it is going to be self-limiting disease. Now, for the identification of Helicobacter pylori, you can see here in this table, we have the test name, we have the invasiveness, how is it going to be carried out, whether it is invasive to the patient or not, and whether it is a reference or not. By reference, we mean gold standard or not gold standard, and whether it is possible to identify any antimicrobial resistance profile doing that test. So from that table, we can tell that the culture, for example, is considered. So what are the gold standards or reference tests? Well, the culture, obviously, isolation of the microorganism, and also demonstration of the organism using the histological special stains. And here we have the warthin-starry stain, which is silver stain, and also Ginza stain. We can see here the spiral shaped bacteria in histology. It's considered invasive. And also, so culture and histology are considered the reference methods to detect H. pylori. And both of them are considered invasive along with rapid urease test. Why is that? Because they are going to be carried out after getting a biopsy from the stomach via the endoscopy. Okay, the oral gastroduodenoscopy, OGD. So by doing the endoscopy, we get the biopsy, and from that biopsy we can do the culture, histology, and rapid urease tests. So these are all considered invasive. Okay. In addition to that, we have the molecular test. It is mentioned as both invasive and non-invasive. Why is that? Because in molecular methods, here they are. We can take the biopsy and we do molecular tests, or we can take saliva, for example, which is non-invasive. Therefore, it is both invasive and non-invasive. It is not considered as reference because detecting the antimicrobial at the genetic level doesn't necessarily mean it is active. And antimicrobial resistance, it is possible to identify those genes responsible for antimicrobial resistance. So for antimicrobial resistance, we have the culture and we have the molecular to identify the profile of resistance. Now, all that what we mentioned is that in the histology is the warthin-starry stain, and this is the stain, warthin-starry stain, and you can see here this is the stomach, a part of the antrum, and these are epithelial cells, and we saw on the mucosal surface the microorganism, those dark stained, and these are the spiral or helical shape microorganisms living in the mucus layer. So they are non-invasive, they are living in the mucus layer. Okay, now this test is the rapid urease test. It is invasive, and if it is pink, it is positive. If yellow, it means it's negative. And we have serology, urea breath test, and stool antigen test. All of these are non-invasive. For the stool antigen test, it can be useful not only for diagnosis, but also for follow-up after eradication. To see whether there is shedding of microorganism or not. If there is continual shedding, it means that there is disintegration of the microorganism and damage or killing of these cells. So this is all about the Helicobacter pylori identification test, and this is the end of our lab session. Thank you for listening.